Hey everyone, in today's lecture we'll be covering plasma membrane in detail and in a very simple and exam oriented way. So plasma membrane is a dynamic fluid structure that forms the external boundary. It is selectively permeable to regulate molecules in and out of the cells. In 1972, Singer and Nicholson proposed the fluid mosaic model. So plasma membranes viewed as quasi-fluid structure. Quasi-fluid means they will partly behave as solid and partly as liquid, in which proteins are inserted into the lipid bilayers. So we have two types of proteins, integral protein and proteins which are present on the surface, peripheral protein, and this is the lipid bilayer. It explains both mosaic arrangement of the protein as well as fluid movement of lipid and protein. So let's talk about the chemical constituents of plasma membrane. It comprises of proteins and lipids. Ratio of protein to lipid depends on the cell type. Suppose we are talking about human RBC. So in human RBC, lipids are 50% and proteins are 49%. Similarly, in mouse liver cell, there are 54% lipid and 46% proteins. Carbohydrate comprise only 5 to 10% of the membrane mass in plasma membrane. They are attached to proteins, either bound to proteins called glycoproteins or bound to lipids called glycolipids. Lipid bilayer composed of two leaflets of amphipathic lipid molecules. Amphipathic means both hydrophobic and hydrophilic. So we have hydrophilic heads and non-polar or hydrophobic tails. Primary physical forces for organizing lipid bilayers are hydrophobic interactions, very important. So there are three types of lipid molecules present in the lipid bilayers: phospholipids, glycolipids and sterols. So let's talk about phospholipids. Phospholipids, there are two types of phospholipids: glycerophospholipids and sphingophospholipids. So glycerophospholipids, they are most numerous in plasma membrane. They are derived from glycerols, contain glycerols, fatty acid, phosphate and alcohol, example choline. So phosphoglyceride or glycerophospholipids, they are two types depending on the alcohol linked to the phosphate group. If alcohol is choline, then they are called phosphatidylcholine, example lecithin. If alcohol is serine, they are called phosphatidylserine. Next is sphingolipid. So in sphingolipids, sphingolipids consist of amino alcohols, sphingosine instead of glycerol. Amino group of sphingosine backbone linked to the fatty acid by an amide bond, example sphingomyelin, and they are not abundant. So over here this is the amino group amide bond which is linked to the fatty acid chain. So this is all for today.